Hello, I'm Theresa LaVallee from the Parker Institute for Cancer Immunotherapy, or PICE. And today I'd like to introduce you quickly to the Parker Institute. It was founded by Sean Parker, entrepreneur and philanthropist, who really had a bold, disruptive idea to bring people together as a collaboration organization with the idea of bringing multidisciplines together as a cross-functional team to work together, breaking down silos with resources and technology to really use the best science, pre-publication, cutting edge, to ask critical questions of how to treat patients with immunotherapy. And our mission is to accelerate with urgency treatments to cancer patients using immunotherapy. The Institute has offices in San Francisco, but collaborates broadly with industry, but also with some of the thought leaders in the immunotherapy space, with the 6 founding institutions shown on the left from the US major medical centers, as well as many other luminaries and thought leaders throughout the field at other institutions, working together as a single team. So all of these people, while many pharma partners have collaborations with different academic centers, this is really thought to be that each of us is part of the PICE team, and there's not the MD Anderson team and the Memorial Sloan Kettering team. We have purposely focused on hard to treat, underserved tumors for immunotherapy, with the idea that we're trying to not be constrained by time to market, pharma prioritization, and really looking at novel science-driven approaches to bring immunotherapy to a variety of cancers that overlap quite nicely with many of the tumors that are being studied for focused ultrasound. We have end-to-end clinical trial capabilities at PICE, in which we look to take science-driven clinical hypotheses to clinical trials. We can sponsor INDs and run clinical trials with our medical centers so that they participate as part of the study team, as well as the pharma partner, and we can use compounds or technologies from multiple different players. So again, we're not bound by portfolio or other constraints that other organizations may find, to look at science-driven clinical hypotheses in these trials. We collect samples, tumor, blood, and stool at baseline and on treatment for each and every patient on the study. And the idea is to answer the question, did the drug work or didn't it work? And most importantly, I think that is not very often looked at is addressing why things don't work, so that you can learn for what was wrong with the clinical hypothesis and refine it. And so in these platform studies, we can very much adapt to adding a new treatment, expanding the cohort in a biomarker selected patient population, or decide that this therapy really isn't effective in this disease type with the knowledge known to date. So the idea is not to just keep doing trials and looking for p-values, but truly to understand why it works, when it works, and why it doesn't work. So this is just showing the PICE translational suite with the multi-omic, multi-parameter analysis that we apply to each and every patient in our clinical studies. So when we say that we have top-line clinical reporting, it includes the biomarkers. It's not just the clinical data, but it's the totality of the data generated in the study. And I think this is really important because the complexity of the immune system is quite large. So when we think about the crosstalk between the tumor and the immune system or the tumor microenvironment, each tumor could be different, and presenting to the T cells by the dendritic cells and the lymph nodes, what's in the periphery, what's the microbiome look like that's influencing immune tone, and really thinking about a multi-component complex interplay between multiple host, tumor, and immune factors to really look at how we can activate anti-tumor immunity. And so we set up, taken from the famous review by Ira Millman and Dan Chen, this a tumor immune cycle to say, what is the clinical hypothesis of the treatment we're doing, and how do we test that to show whether it's working or not working? And while we're always focused on the T cell for either checkpoint inhibitors or cell therapy treatment, that many other cell types are of critical importance to have a productive immune system. And so while combination with PD-1, PD-L1 agents has been very successful, it's not the only answer, that there are many other treatment approaches that need to be looked at for activating anti-tumor immunity, and understanding when PD-1 is the predominant mechanism in an antigen-experienced, quiescent, immunosuppressed T cell that just needs to be reinvigorated, or are there other components of the immune system, such as the presentation of antigens, or could it be super suppression from a lot of macrophages or Tregs? And is that what you have to overcome to really activate that anti-tumor immunity? So really thinking broadly about the immune system as a multi-component system, and also really looking at the tumor as influencing the immune cells that are within the tumor microenvironment. And so when we look at the objectives of this conference, I love it because it's very well stated to really think about what are current challenges and potential future research directions for focused ultrasound immune-based treatment for cancer. But then taking a step back and saying, what focused ultrasound? So maybe we need to be more specific about the type of focused ultrasound that we're using, because it would not be unreasonable to assume that each of these has a different immune mechanism and influence on the tumor and immune cells. So define the relevant knowledge gaps. To me, there's a lot of beautiful work being done within the network of collaborators within the Focus Ultrasound Foundation, and work that they and CRI have supported to look at activation of T cells, dendritic cells, B cells, and publications that show that, but really to get into which type of focus ultrasound is affecting which cell types and how. What is it activating in the immune system? What's missing in the immune system? What I like to say when we study tumors such as pancreatic cancer, what's broken in pancreatic cancer that we need to add a therapy to then drive that immune cell system in? Do we need to get more T cells in the tumor? Do we need to get rid of the stroma? Do we need to get rid of the myelosuppression? So all of those things to think about how to design treatments to really look at how to complement combinations with focus ultrasound, and which type goes with which drugs and which tumors. So I very much look forward to the discussions, and thank you for the opportunity to present.